So now, too bad that we can't go back to the labs at this moment to use the microscope. We know that I can actually point out each part of the microscope and we can have a learn to learn this. Okay, so but generally we can learn it through the diagram first. The microscopy is a technical field using the microscope to view the object and the area of the object that cannot be seen with a naked eye. Okay, so that's why I use microscope. So a microscope is an instrument that magnifies the object, produce an image in which the object appears larger. So this kind of the image is known as micrograph. Okay. So we started with the light microscope. So modern compound microscope feature a two-stage magnifying design built around a separate lens system known as objective and eyepiece. So the one that mount onto this tube, okay, is the body tube. So the lens, okay, the lens near to our eyes is known as the eyepiece, and the lens actually near to the object is called as objective lens. Okay, so we do have the holder, we do have the arm and the head of the microscope, switches, okay, and then we have two knobs to adjust. Okay, one is to adjust the intensity of the light, another one is to adjust the focus. So we have the coarse and the fine knobs, and then we have the light source or illuminator. So illuminators will be focused onto our specimen where we need to clip our specimen. So by using the clip, we place it on the stage. So it means that the light will pass through the specimen, the thin specimen, and then enter into the objective and then reflects, okay, reflects and then enter into our eyes. Okay, so generally these are the components of the light microscope. Okay, now basically in your syllabus you're not required to actually remember. This is your, I mean, the IGCSE, your SPM classes. Maybe you need to learn how to label. This is just a quick revision, because if I don't tell you guys the name, you guys do not know the name. So when come to practical class, and when I tell you guys, okay, please hold the arm, okay, this turn, okay, your objective lens, you do not know where. Okay, so that's why we need to learn the parts of the microscopes. Okay, now, so first of all, we look at this, the lens that near to our eyes. The lens that near to our eyes is termed as eyepiece. Okay, so the lens that's near to the object, okay, the lens that near to the object is termed as the objective lens. Okay, objective lens, and we do have something to hold. Can you see that, the grid, okay, of the objective lens? So how you move this objective lens, because we have three of them, three different magnification. When you move it, always remember you move using the grips here. So this part is known as the nose piece. Okay, the holder here is called as called as nose piece. It's the part where we're going to adjust. So don't touch on the objective lens. Yeah, don't move by using. Don't touch on the objective lens. You only move your. Okay, let's say for example, I want to change from yellow to blue color lens. Okay, yellow to blue color lens. I have to move this part. Okay, can you see that highlight? Okay, so now you can see that. Okay, we have the tubes here. I need to combine to link it from the eyepiece to the objective lens. We link it, so we known as the tubes or the head of the microscope. So sometimes I would say that, okay, you can turn. Okay, basically we have the screw here. You have the screw here. So this screw can allow us to turn our microscope heads. Okay, when you store them. Okay, so this part is flexible. It's not like fixed. You can turn them. Okay, 180 degree you turn, and then you make them a little bit compact in shape, and you can actually store them nicely in the cabinet. Okay, and the parts that you hold, very important. Then you hold the microscope. So this part is known as the frame or arm. Okay, either frame or arm. Okay. Next, you can see that the place that we place our specimen, okay, of slides in this case is termed as the stage, and we need to clip them, right? We need to clip them. So this is the specimen holder, okay, or specimen clipper, okay. So when you hold the specimen here already, we need to adjust the positions, right? You move left, right, front, back, right? You need to adjust the stage. So we do have these adjustment knobs here. we call mechanical adjustment knobs or adjustments to allow the stage to move. So answer is mechanical stage control that allow us to move the stage left, right, okay, front, back, okay. Now, in order to make our stage going up or down, right, we need to focus, right? So we need to have the focusing knobs here. So we have the large one and a small one, okay? Can you see that? Large and the small. Can you see that? Large and small, okay? So we have the large one is termed as a coarse focusing knob or focus knob, and we have the fine focus knob. So I will teach you guys how to use it when we go back to the labs, okay? And next, definitely we want to have the light, correct or not? The illuminator of the light source here. Okay, illuminator or light source. And then when the light, we need to focus the light onto our specimen, correct or not? We need to focus. So this part is known as the condenser. So condenser will be the one that actually focus the light. Okay, so next, because the light intensity here, we need to adjust the light intensity. So we do have the light intensity control knob as well as the switch, okay? Because after we finish using it, we need to switch it on or switch it off, right? So we have the light switch at the side. So understand the name of each component. So when in the class or practical class, I can tell you, oh, please look at the light switch. Okay, hey, you forgot to switch your light switch, and then you don't tell me, hey, where is the light switch? Then too bad, okay? You need to know, okay? You need to know how to find that particular switch or particular knot, okay, yeah? So next, assume that you guys have done this one as well. So let us look at this. So for a cross, so what is the light source for a microscope? Number 2, what is the light source of a microscope? We call it illuminator, okay? Illuminator. Okay, second. So what focus the light on the specimen under the observation? It present give the sharper image. Number 5, what's this answer? Condenser, okay? Condenser. Okay, next. So which focus knob is the large knob which move the stage to produce a large change in the focus? So we have coarse adjustment knobs or coarse focus knobs. Okay. So what is the lens? So what is the things that connect the eyepiece to the objective lens? We call it the nose. Okay, nose. Or connect eyepiece to the objective lens. Okay, it's the arm. Okay. And then next, or the head, or the head also can. Okay, the head or the arm. Okay, next, what is the lens that you look through at the top of the microscope? The top of the microscope. So which is termed as eyepiece. Okay, eyepiece. Okay, next, what is the name for the knobs? Okay, which is small in size, which move the stage to produce a small change in the focus. Okay, so down, so fine adjustment knobs. Okay, fine adjustment knob. So the holder of the different objective lens, the holder of different objective lens is known as the nose piece. Generally, we have 3 to 4 objective lenses, okay, are found on the microscope. So where we place the specimens is known as the stage. Do you get this correctly? Okay, so you know that the functions of each of the components of the microscope, right? Okay, any more questions from you guys? No, then I stop recording.